And this is the pre-lab webinar about alcohol dehydration. We'll run through this pretty quickly. It's a fairly easy reaction. It's going to demonstrate the E1 mechanism, which we haven't talked about yet, and it follows in conjunction with this series of lectures that we've had on nucleophilic substitution as we move through the different kinds of substitution and elimination reactions that go from SN1, SN2, E1 to E2, and then how we all correlate that together. Again, this will specifically focus on the E1 mechanism. In the general overview, alcohol dehydration involves using an acid to promote an E1 mechanism through the removal of water using heat. The use of the heat helps to force the product formation by removing the product as it forms through distillation into, in this case, in this experiment, we'll be using a Hickman still head to collect the distillate that you remove. And so over time, as you remove the product, you force more of it to form. This will tend to yield many products, depending on the substrate that you're using. Sometimes you don't have as many as others, but in general, we tend to give substrates that may have anywhere from 2 to 4 products that form, at least with this experiment. And the key to that is remembering that you have to walk through the mechanism, which involves the use of a carbocation. And so each time that you are given a substrate for this reaction, you need to be sure that you go through and actually draw out a mechanism on paper and think about all the different products that are possible to be formed from what is going to happen. So in the case of alcohol dehydration, the water that has the acid in it, which is usually phosphoric acid because it helps with the dehydrations, the proton from that will go to the alcohol molecule and make it into a water molecule that makes it a better leaving group. And so you have to walk through that. You have to see where that's going to leave and then leave the carbocation present there. And once you have the carbocation, you have to think about what would happen if it removes the nearest hydrogen and forms a product, or what might happen if there was a methyl shift or a hydrogen shift. You have to think about all those different possibilities. And you have to remember that the molecule in question wants to form the most stable arrangement. And so by following those rules, you will arrive at both the major and the minor products that will occur. The major product, again, is generally the one that has the most substitution on either side of the double bond that would form. So in this case, for this particular species, this was the major product, whereas this was the minor product, because even though it was possible, and even though this was possible, they're not as favorable as this product. So these two are minor products. The apparatus we'll use Monday is fairly simple. It involves a heating plate and an aluminum heating block. We use the aluminum heating block when we use the microscale kit. The microscale kit has the Hickman still head, which is what we will use for this experiment. If the alcohol is a low boiling alcohol, which should be designated on the standard lab page, then this form of the apparatus is used, where a Claisen head adapter is placed in between the Hickman still head and the reaction vial. If it is a high boiling alcohol, then this setup that is just the reaction vial with the Hickman still head is used. Though these pictures are from one of our older slide sets, it is important to remember that you will need to follow the instructions in the lab manual with regards to how to place the lollipop wand thermometers through these apparatuses to measure temperature, should temperature measurement be required. Sometimes we don't always have to use a thermometer. If it depends on what you're working with. Like if you know most of the product that you're going to form is going to boil up into the top, sometimes you don't bother with the thermometer and you just collect it. But it's a better practice to use the thermometer because, again, you want to make sure that you're actually collecting over the temperature that is relevant to the products that you would like to collect for the most part. And as far as analyzing this particular set of products, because it's all going to be a liquid, there's not going to be anything from these particular sets of products that's going to probably end up being a solid. Because of that, we have to use other methods to test the purity and the composition of the samples. And this is where gas chromatography is useful, because we can inject a small amount of the sample using a syringe into an analyzer that has a gas chromatography column, and based on retention time, which generally the time it takes for it to go through the column is what we would view retention time as for a gas chromatography. But what you have to remember too is that gas chromatography is based off of the boiling point of the material. So the higher boiling the material, the longer it's going to take for it to go through the column, because the lower boiling is going to come off first. So it's a lot like distillation in that regard. And so typically the alkenes are going to have a lower boiling point than the alcohol that you started with. So unreacted material tends to be the last peak. And then major products and minor products, that will depend on the boiling points you can find for those possible substrates. Sometimes you can find them, sometimes you have difficulty finding them. You just have to try the best you can to find them. Another way to tell would be by how much of it is produced. And so you could, base it off of that information too. And then sometimes you have contaminants or noise, and we'll show you kind of a picture of that. So when you're looking at these gas chromatography printouts that you'll get, they'll actually look a whole lot better than this, because we've been using more computerized samples lately instead of the older ones. We used to have older ones that used a strip chart. Now we have a newer machine that's got a robotic auto sampler, and they usually just run all of them on those, and they have a set, and then they'll put some instructions and assign it on iLearn, and you can just go and you have one to interpret. And that is better for some reasons we'll talk about. But essentially, you just want to look at this line here as if it's an intensity from an instrument. It's some sort of reading the instrument's giving, and it's just plotting it here. There's not really necessarily a unit that's always associated with this. It's just kind of an intensity. And then the time is what would be on your x-axis. So you're looking at it as it crosses over across time. And then if you look here, you have your GC peaks that are occurring, and this one would be probably my major product because I have what appears to be the most of it concentrating there. Then I have a minor product that's over here and another minor product here, and this is going to be my unreacted material most likely. And I actually drew this for a specific reason. This is one of the more common GC printouts that I see with students and there are experiments where you'll have some of the products you wanted to make, but you also have quite a bit of unreacted alcohol that's still in the sample. And then over here, this would be what you would call signal-to-noise ratio perturbance, or just noise. It's not really necessarily a peak. It could be a contaminant in some cases, but that's probably just noise from the instrument. All instruments have a little bit of noise, and the key is to do enough statistics so that you minimize the noise and increase your signal. Now, if you were to be given a strip chart, instead of being given a report printout that gave you the area under the curve, which is how you determine the mole relationships of what is present in the sample. So we can correlate the area under these curves as to the molar relationship in the sample mixture. And we do that by taking it as a fraction of the total areas under the curve versus the area under the specific curve. So instead of using mole, you use the area. So area over total area is the mole fraction for that particular compound in the sample. And you can compare that to theoretical yield if you want. Because you can work through a reaction. If you know what your limiting reagent is, you can figure out how many moles theoretically you should have of each product, and you could figure out, if it was a mixture, what the molar fraction should be for each compound. So you can actually measure whether or not you're getting close to that by looking at the mole fractions. On newer instruments, you don't have strip charts that have these curves like this. So that is the advantage, in that they will integrate the area under the curve for you and print it into a table at the bottom, or even on the computer screen, and you can write it down. And it's much more accurate that way, because obviously these can take on different shapes. They may not be a perfect square or a perfect rectangle, which is like a really easy shape to calculate the area of. It's probably something a little bit curvy and has little bitty infinitesimal changes, so you have to integrate that to actually get the area that's really there. But in the old way of doing it, what you would do is you would look at the curve, and in most cases it takes on the shape sort of like a triangle. So you would try to find the midpoint of the peak and measure the height, and then you could measure the width at the bottom, and you could use something like the area for a triangle equation to calculate or estimate what the area under that curve might be. Another way of approximating it would be to draw a small rectangle that encompassed the peak and ignore the space in between, and so it would have a little bit of error, but it would be kind of a rough approximation. So those are different kinds of ways of doing it when you have something like that. But the best way really is to integrate it, and to go from the point where the curve begins and the point where it ends, and integrate it with respect to the distance on the bottom, which would be in time in this case. And so it would simply be this sort of linear relationship here, where you're just getting all these rectangles under here. That would be for that triangular looking kind of curve. Again, it's not a perfect system when you do it that way, and that's why the newer computer models are more efficient.